Thank you for joining me this morning in what is a new event for the Academy, our President's Breakfast. It's a wonderful opportunity for us all to meet in an informal setting and to get to know our outstanding 2024 new Fellows a little better. Each of our Fellows was elected for their incredible and ongoing contributions to health and medical sciences, and this year's cohort is no exception. Joining us are experts in social work, pharmaceutical development, First Nations health, obstetrics, paediatrics, infectious diseases, physiology, and many more disciplines. Their wealth of experience is tremendously valued by the Academy, and we look forward to their input into our activities. Many of our new Fellows are pioneers in their field, have made seminal discoveries and have made a significant difference to health and medical research, not just in Australia, but clearly internationally. Their accomplishments are hard to summarise, so of course we're challenging them to recap their achievements in front of you all today in just 3 minutes. Each of our new Fellows joining us today have boldly taken on the tricky challenge of presenting themselves and their work in 3 minutes in a series of rapid-fire talks. Similar to the famous 3-minute thesis competition, we've asked Fellows to speak on a topic of their choosing, their research, their proudest achievements, and so on, for only 3 minutes. In fact, our Secretariat will be keeping them to time with a bell. It's a difficult task for a PhD student. One of my PhD students earlier this week was worrying about how she was going to do a 5-minute talk when she's only used to talking about the same topic in 20 minutes. And she's only just been a student for 2 years. But let alone for our Fellows with their numerous publications, accolades and discoveries. Keeping to the spirit of the event, I encourage you all to applaud their bravery and take the time to congratulate them on their efforts and ask questions about their work during the day. We have 20 Fellows speaking this morning, and we'll take a break after the first 10 to allow everyone to partake in the refreshments. So please welcome our first speaker, Adjunct Professor Craig Rayner. Thank you, Craig. It starts from when you're up here.
Great. So look, I'm the first of a family to get beyond high school, so being here is pretty cool. And I'd really just like to acknowledge the Academy and also Glenn Begley for spearheading the application. I'm really chuffed. So my career, I've kind of spun around the world. I've had senior global roles at R&D companies like Moderna, Roche, CSL. I've also founded companies at the cutting edge of technology-enabled medicine development, including D3 Medicine and Certara. I've also held clinical and academic roles, and I'm currently the Director of Translational Medicine in Infectious Diseases at Moderna, as well as an adjunct research professor and distinguished alumnus at Monash. I think in all my roles, my focus has really been on leveraging science and technology to accelerate patient access to medicines. It's clearly a team sport, and today I'll just briefly outline impact across 4 key areas, that being patients, innovation, policy and ecosystem, and business. So for patients, I've been really fortunate to contribute to the approval of several important medicines, including linezolid for multi-drug-resistant infections, Tamiflu for infant and pandemic flu, moxidectin for river blindness, and played a key role also in identifying and debunking potential COVID treatments. On the innovation front, I've published over 120 papers, including in top-tier journals like New England and Lancet, particularly on model-informed drug development and novel clinical trial designs, including the 5th most downloaded article in New England Journal History. At the policy level, I've had the privilege to work globally, advising organisations like WHO, developing curricula for FDA, chairing sessions at the UN, and supporting medical countermeasures and preparedness and COVID response for the Australian government. In business, my experience is serving as the Global Due Diligence Director at Roche, advising on many asset and licensing acquisitions, founding and being a CEO of D3 Medicine, and President at Certara, supporting it through its $768 million IPO and getting it to a market cap of over $7 billion US. I'm really excited to join the Academy, and I look forward to contributing my multi-sectoral experience to advance contemporary medical product development and regulatory science for the betterment of Australia. Thank you.
What a great start. Thank you, Craig. Can I just make a comment? I've been struck by talking to different people in our fellowship over the last few years. Just actually quite a number of our fellows are people who are either the first in their family to go to university or have similar sorts of stories. It's quite fascinating. Anyway, we'll move on now. I'd now like to introduce Professor Kim Delbaere from Neuroscience Research Australia. Thank you, Kim.
Thanks, Louise. I'm also very honoured to be here, and just want to point out that I started as a mentee, so other mentees here. It's a good way to get to know the Academy. I'm a physiotherapist by background. I'm a senior principal research scientist at Neuroscience Research Australia, which is a medical research institute affiliated with UNSW. And my journey in health science has really been driven by a deep commitment to improve the lives of older people. My focus is on healthy aging, fall prevention and recovery. And at the core of my work really is a desire to empower people, older people, to stay active in every way, so physically, socially and mentally, and through that to preserve their quality of life and their independence as they age. And I thought maybe I'll share a few statistics so that you understand kind of what the issue is, as we always have to talk about that. But we all know that we have a population that is aging, and we are actually now at the stage where we have more older people, as in 65 plus, than children younger than 16. And that's the first time in history that that has happened. And we've got a huge societal impact coming onto us in that way that we, in our health systems, need to prepare for. And another interesting stat that we are having a collaboration with Rotary, and they came up with this statistic, which is very striking. Falls in older people is very common. 1 in 3 older people above 65 will fall once a year. But we also have 16 people who will die a day because of a fall, and that is the same as melanoma, drowning, suicide, cycling and car accidents combined. So that is a statistic that we actually don't often hear about. So I've embraced digital health solutions to really come to address some of these issues, and we have developed a digital health app to prevent falls and to have the reach to older people in the community, where we have been able to show that we can prevent falls by 42% in high-risk groups of people from using the exercise program for 6 months and longer. We are very much at heart, we have all equity in relation to both culturally and linguistically diverse communities, as well as Aboriginal and Torres Strait Islander communities. And we really hope that we can develop more of these tools to really address the reach and make sure that we can prevent falls and support healthy aging across the community. So through my work, I really aim to create a world where aging is not a barrier, and to have a full and a fulfilling life for everyone.
Thank you, Kim. I certainly listened with great interest for a personal reason for that. Our next speaker is Professor Justin Gooding from the University of New South Wales. Thank you, Justin.
Thank you, and firstly I'd also like to say I'm incredibly honoured to become a fellow of this academy, and I'd like to acknowledge Maria Cavalaris, who was the person that nominated me after I kicked and screamed for a few years about wanting to do it. I'm a chemist, and so what my research team does is modify surfaces with a single molecule thick film. And why that's so important is because we can then determine how that material interacts with the outside world. And we can bring multiple different chemical functionalities so that the material can interact with that outside world with all the different aspects of what that outside world is like, and yet use the same bulk properties of the material. So what has this got to do with biomedical science? Well, the outside world that we interact with is biological mediums. And so then this becomes applicable for anything from a diagnostic device to a biomaterial, to a tool to allow you people to understand fundamental biological or biomedical processes. And so what we really do is develop tools so that the community out here can do the important science and the important clinical work that they do. So some examples of this from our own work is we've been involved, my first PhD student commercialised a glucose meter, developed a company called Agamatrix, which is a $200 million a year company that we're involved with from the very beginning. You don't know the name of the company, but you might know the device. They developed the first glucose meter that interfaced with an iPhone. They actually convinced Steve Jobs to allow Apple products to be used for biomedical devices, and it's the first biomedical device ever to be sold in Apple stores worldwide. It may have led to them thinking about the Apple Watch. We don't know that. I also developed, and of course, glucose, the most used analytical method in the world, 10 billion assays done a year, 10 times more than anything else, which is the pH meter. We also developed a 3D bioprinter with Maria and a company called Inventia Life Science that allows you to make 3D cell cultures and high throughput, 3D organoids. And we're now using that to try and understand the outcomes from a liquid biopsy using some other sensors. And probably the biggest thing we're working on at the moment is a patch that goes on the arm, monitors drugs in the interstitial fluid in real time with a company called Neutromics. That means you could then keep the drug, the first drug is vancomycin, in the therapeutic range and avoid toxic effects. The beauty of it is we can get reversible signals so we can continuously monitor. We think that's as big a game changer as the glucose meter was for diabetics. Thank you.
So much diversity in what people are doing. How fascinating all of this is. We come to our next person, Professor Steve Wilton, who's from Murdoch University. Thank you, Steve.
Also, like the others, thank you so much for this great honour. It's quite fitting. I actually did a PhD in Adelaide University biochemistry in 1980 or something. With George Rogers. It was cloning and sequencing of keratin genes. I then started working for a couple of small biotech companies. One of them is still going. It was a spin-out from the biochem department. It's called GeneWorks now, so it's been going for over 40 years. The other was a company in Western Australia, and that's where I learnt PCR, the DNA amplification technique, the hard way. I was actually making the Taq polymerase, making the primers, and designing the applications, whether it was fish, fungi, or human genes. I started working with Nigel Lang at the Australian Neuromuscular Research Institute, and what I was tasked with there is finding the mutations that were causing Duchenne muscular dystrophy. Now, Duchenne is the most common and serious form of childhood muscle wasting. It's a big gene, it's an accident waiting to happen. There are spelling errors in the gene that stop it from working. Kids are in a wheelchair before they're 12, and at that time, most would be dead before they were 20. I came up with this really crazy idea to use antisense oligos. These are small synthetic gene patches that would target the defective part of the gene message, and give the cell the ability to spit the bad bit out and make a functional message. We now have 3 FDA-approved drugs to treat Duchenne muscular dystrophy. The compounds were licensed through the University of Western Australia to a company called Sarepta Therapeutics in Cambridge, Massachusetts. So I get to go to Boston a couple of times a year. I'm actually catching up with the Sarepta folk tonight and tomorrow and the next day. There's a big conference in Perth, so I'm back there for this. So this is a $1.2 billion market last year. I never thought you could commercialise a rare disease, but it obviously has really taken off. No worries. What we're doing now is taking this technology and applying it as personalised medicines. So with a group of colleagues around Australia, clinicians, all the states, all the capitals. We're looking at trying to industrialise personalised medicine. So we have a pipeline that will find amenable patients to these gene patches and then get the drug made, tested and supplied to the clinician to treat their patient. Thank you.
Thank you, Steve. We now move on, and I'm very pleased to introduce Professor Scott Bell from the Translational Research Institute. Thank you, Scott.
Thank you, Louise, and thank you to the Academy also for this honour. Last Saturday, Harry Coffey rode an incredible race and won the Caulfield Cup. On the same day, he was 29 years of age, and he was born with cystic fibrosis. His life with advanced lung disease required frequent hospitalisations. When he was born, the median age of the CF population was 14. It's now greater than his current age. Many factors have contributed to that improvement and the doubling of survival during my working life. This has resulted in a trebling of the number of adults, impacting on the burgeoning adult cystic fibrosis centres, and there have been some unexpected consequences. Cross-infection between people with cystic fibrosis is common with the common CF pathogens. I have been privileged to lead the CF Air Group since 2000, a large interdisciplinary team. Our first set of work identified genetically indistinguishable strains of Pseudomonas aeruginosa across the CF population, particularly the adults, and this was associated with more adverse clinical outcomes. Of course, wisdom, or perhaps dogma, at the time was these bacteria were held in droplets greater than the size that were easily respirable and were unlikely to travel more than 1 metre. This conflict between what appeared to be cross-infection, person-to-person spread of bacteria, and the dogma led us to set out to try and better understand mechanisms. We identified potentially infectious aerosols less than 5 microns in diameter, easily within the respirable range, that travelled for more than 4 metres from their source of coughing and remained airborne for more than 45 minutes. During the period of 2019 and before 2020, we sought to examine mitigating factors, options such as changing ventilation in our hospitals to reduce risk, and we studied the role of face masks before they were a thing. And we found them to be highly effective and impacted on CF infection guidelines and policies globally. Sound familiar? Our current work is examining environmental links that might lead to the acquisition of common bacteria, particularly in an ever-changing built environment. Finally, I'd like to acknowledge the consumer involvement for 25 years, both in clinical trials and in understanding cross-infection in cystic fibrosis. Thank you.
Thank you, Scott. And we move along. Our next speaker is Professor Linda Denehy from the University of Melbourne. Thank you, Linda.
Thanks, Louise, and I too would like to say I'm very honoured to be here and like to thank Professor Karen Thursky for nominating me and the support from Peter Mack that I've had. It's been fantastic. I'm a physiotherapist and I started my career as a clinician in a quite large regional centre near the Victorian ski fields. Yes, I love skiing. All physios seemed to travel, and off I went, and I spent quite a lot of time travelling Europe, ended up working at a hospital called the Brompton Hospital in London. The Brompton Hospital has one of the world's leading cardiorespiratory hospitals, so all patients are cardiac or lung patients. Many of you might wonder what a physiotherapist does in these areas, when our most known role is really musculoskeletal physio or running onto the Adelaide Oval, really, and helping the footballers. Cardiorespiratory physio involves both acute treatments as well as rehab. So, for example, managing people in intensive care, getting people up and about after major surgery, and providing rehabilitation for people with lung disease and heart disease. My time in London really stimulated my interest in research because I was involved in research at the Brompton. And I joined the University of Melbourne to teach physio students and undertake my PhD all at the same time. That was hard. I remain a teaching and research academic. I had a bit of a sojourn for 12 years in management in the faculty, but through these times I've maintained my enthusiasm and commitment to research and research mentorship. So I'd like to share a few highlights of my research. The first is that I, along with a lot of my clinical colleagues, saw a gap in physio management of patients in critical care. Patient deconditioning, while intubated, ventilated and bed-bound, was really not being addressed at all. So we developed the first NHMRC-funded Australian trial, physiotherapist-led, in rehabilitation in critical care in 2008 to address this significant functional decline. This also led to work in working with patients with tetanus in Ho Chi Minh City through a Wellcome Trust grant, which was an amazing experience and very fulfilling for me as a person and a physio. I joined Peter Mac as a joint position, and to build capacity in research, and I was really immersed in prehabilitation as well as rehabilitation. So prehabilitation for people before their cancer treatments to really optimise their physical and mental state, and really try and help them, empower them to be part of their journey, and worked with many consumers in this role. We currently have funding along with RPA in Sydney for 3 MRFF grants to look at digitally enabled prehabilitation. Finally, I'm a passionate mentor, and I've mentored many physio PhDs in allied health, and I've watched them grow with great pride as they became successful clinician scientists. Thank you.
Thank you. We move on now, and I want to welcome Professor Deborah Lupton from the University of New South Wales. Thank you, Deborah.
Thank you. Good morning, everyone. It's great to be here. So I'm a bit of a rare beast at this gathering, I suppose. I'm a social researcher. I'm the kind of kid who at high school really excelled and loved the humanities, English, history, art. But I also got really interested in biology and studied it till year 12. So when I went to university, I did play or toy with the idea of studying medicine, but my experience of dissecting a rat in biology in year 12 led me to realise I'm very squeamish. I hate the sight of blood and internal organs and whatnot, and I just thought, no, I just can't cope with being a medical student and actually practising medicine. So what I decided to do is actually study biological anthropology and sociology, and particularly health sociology I got really fascinated in. So I did those. I did that training at ANU at honours level. Then I went to Sydney University and did a Master of Public Health. So I learnt about epidemiology, biostatistics, health promotion, health economics. And I stayed on in that department to do my PhD. My PhD brought together an interest in the way that health is communicated through the news media. So it looked at news reports of HIV and AIDS from the first news report in Australia right through to 1990. So it was a bit of a social history of how the news media had covered AIDS in Australia. Then I got a job in health communication, teaching health communication to communication students. And I've sort of since then gone back and forth between sociology, communication, sometimes public health, and combining those those disparate interests. I now work at UNSW in the Centre for Social Research in Health and the Social Policy Research Centre. So I get to use, again, all those interests and expertise to look at the social aspects of many conditions. I'm very interested in creative and qualitative research methods. I brought along this little booklet that I published recently. It can be found online, or I do have a few copies with me. I'm really interested in how we can take our research findings and communicate them to the public through exhibitions, through films, through artworks. And so that's one thing I'm really super interested in continuing on, is how to take interesting research findings from medicine, from public health, and communicate them to publics. I'm very interested in fresh air and air quality. I've got my CO2 monitor with me, so I'm able to monitor. It's actually quite good in this room at the moment, by the way, you'll be glad to hear. And I'm also super interested in planetary health and digital health and bringing those two quite disparate but interrelated things together. Thank you very much.
Thanks, Deborah. And you can see how Deborah's interests overlap with so much of what we discussed yesterday. Just a general comment, Deborah is one of our fellows who belongs to one of the other 4 Learned Academies. And in fact, we have fellows in each of the Learned Academies, so Humanities, Social Sciences, Technological Sciences and Engineering, and Academy of Sciences. I think we might be the only academy that has fellows across all of them. Shows you the breadth of health and medicine. Thank you. We now move on to our next speaker, Anthony Weiss from the University of Sydney. Thank you.
Thank you very much, Louise, and thank you also to the Academy. It's just an absolute joy, and I've been so enjoying speaking with members of the Academy. It just blows me away. It's a fantastic group. You can also see, by the way, from my photograph, that I'm clearly drawn to the lab, and just by the angle on the shot there. And so I'd like to... clearly doing an ancient Greek pose there, I think, statue style. So I'd like to describe to you, of course, the work that I've been involved in. I too, like a number of other people, am the first in my family to have gone beyond high school. And so I have been naturally drawn, as you can see, towards research. So the question, of course, is what is the research area that gets me up in the morning, gets me excited? And that is the area of elastic tissue. And many of you know what elastin is, but let me just briefly describe it to you. Elastin fundamentally is the building block for a whole range of different tissues. So, for example, as we breathe in and out, we need elasticity for the lung. And as you know, damage to elastin is absolutely awful when it happens. And I won't go through the specifics. But, of course, you can also appreciate that when, let's say, skin is damaged, the elastin within the skin, we lose a lot of skin elasticity. When, for example, we look at blood vessels that expand and contract, we smooth out the peaks and troughs of systole and diastole by means of that elastin as well. And of course, for those of you who had a drink this morning, you'd appreciate the elasticity of the bladder. And so fundamentally then, elastin is pervasive throughout the body, but you might not have known that the major building block for elastin, which is called tropoelastin, just like the bricks in the wall used to build a wall, that major building block is encoded by just one gene. And so basically, therefore, there's this amazing diversity, excuse me, this amazing diversity of how tropoelastin gets used in many different locations in the body. So what we've done has been to effectively make large amounts of tropoelastin, discover how its rules of assembly operate. We make now fibre sheets and tubes. We've used this material to be able to repair a range of different tissues. For example, halve the time it takes for skin wounds to heal up. We have been able to make substitute vessels that can be used in implantation, so their vision one day is that someone would see that someone needs, for example, a bypass, and would take one of these manufactured vessels off the shelf and implant them. And we're already seeing colonisation and conversion to neo vessels under these conditions, which I think is very exciting. By means of one final example, for example, with James Chong, one of the many Jameses last night who won an award. We've been working on LAD ligation, and she demonstrated that we can preserve function of part of the heart with a single injection of tropoelastin, and that's been published as well. So commercialising technology has proceeded very nicely, and let me just thank the Academy again for the wonderful opportunity to be a member here and to present this today. Thank you all.
Thank you so much. Our next speaker, we heard yesterday in a panel session, Professor Dawn Bessarab. Oh, there you are, great.
Well, that was a surprise. I thought I was later. But anyway, it's better late than never. Yeah, right. Okay, who am I? Well, I grew up in Broome. I'm a Kimberley kid. I was the first in my family to enter university. I was a mature age student. I didn't really know what I wanted to be when I grew up. And in Broome, Aboriginal students never really got spoken to about possible career paths. And I didn't find out until I left much later in life. So I graduated with my PhD and then fell into health research. And I'm a social researcher, I'm a social worker, very interested in Indigenous knowledge systems. That's what my PhD focused on. And from the PhD, I developed a research process called yarning. And yarning first wasn't actually my idea. It was introduced to me by a Noongar elder in Perth when I was working in child protection. And what I liked about yarning was that it provided a different way to engage with Aboriginal people, which wasn't threatening, used the process of narrative, storying, and enabled you to have really good conversations with people on very sensitive and delicate topics such as child abuse. So it was a way to engage in really serious discussions, but in an informal way that was culturally friendly. As a result, yarning's become recognised both nationally and internationally, and sometimes I think I've created a beast, but I think what's really important about yarning is that research yarning has gone on, and we've now co-developed with Ivan Lynn, who's a physiotherapist based at Wacker in Geraldton. We've worked on developing the clinical yarning framework, which is having a really good impact and reach into the health profession for doctors to engage differently with Aboriginal people rather than the straightforward clinical or medical interview, to apply yarning, which has better results with our Aboriginal patients and clients in terms of understanding what their health condition is, and you get it through narrative. So the questions that you might ask in a straightforward interview, which you would not get responses to, you would find when someone tells their story, for instance, you might ask them, have you been drinking? No, I haven't. But when you ask them to tell you about what's been going on, they might say, and this is evidence that we've got from research, well, actually, my relative came to town last week and I hadn't seen him for a long time. We went down the bottle shop and got a carton. And it's like, but you just told me you hadn't been drinking. So yarning reveals information that a straightforward interview does not. So social yarning we've used, now introducing that into medical research, particularly for those that are doing surveys, etc. Social yarn helps you to build a relationship with the client before you actually ask the questions. So I guess in terms of social research, applying Indigenous knowledge systems to what we do, my expertise is in culture, cultural knowledge, Indigenous knowledge, and I've mentored and still am mentoring both Indigenous, non-Indigenous researchers, students, in how to apply and do more culturally appropriate Indigenous research using yarning. Thank you.
Thank you, Dawn. It was actually really exciting for me to learn that Dawn was the person who actually really promulgated this concept of research yarning, because I've had a student who used that and I hadn't quite appreciated until yesterday that Dawn was the person who started. So it's always fantastic. We come to the last in this first session. Dr. Christopher Burns is from Amplia Therapeutics, one of our industry-related fellows. Thank you, Chris.
Thank you, and like everyone else, I want to thank the Academy. It's a real honour to be a fellow, and thank Michael Parker, who was my nominator. I'm a chemist by training, and it's great to have another chemist here in Justin Gooding. And my career has all been about drug discovery and development, particularly as it relates to small molecules and particularly in oncology. I've been involved in the discovery of a number of drugs that have gone into the clinic, NK1 antagonists for coughing, an orally active vascular disrupting agent for glioma, and probably most notably JAK1, JAK2 inhibitor by the name of momelotinib that was recently approved by the FDA. It's approved by the EMA, Japan, and hopefully soon here in Australia. I discovered that drug along with Andrew Wilkes, and this year we were co-recipients of the PM's Prize for Innovation for that discovery. I'm really passionate about translating drug discovery and development, and translating the great science we do here in Australia into products that improve patient outcomes. And doing that with a commercial lens. And the other reason is really to help create an industry that will employ many of the graduates and postgraduates that come out of our great universities who may not want to be academics. And believe it, there are some who don't want to go down that path. To that end, I'm on the advisory board of a number of sort of commercialisation incubators, MedChem Australia, Breakthrough Victoria, to name two. In 2016, I co-founded a company called Amplia Therapeutics, and the goal of that company was to commercialise some drugs that were discovered at the Cancer Therapeutics Cooperative Research Centre. Amplia Therapeutics is a company that's listed on the ASX, so everything I tell you now is already in the public domain. We have our lead drug in a phase 2 trial in pancreatic cancer. We think the drug has opportunity in a whole range of solid tumours. We're really excited about the potential there. Using the knowledge I've gained through my career and through what I'm doing now, I hope to bring some of that to discussions here at the Academy. So thank you again.